Good morning. Today I'm going to speak about pathology of tuberculosis. Now, tuberculosis is a disease that has killed probably more human beings than any other disease, maybe excepting malaria. It has been called the captain of all the men of death by the poet John Bunyan. Throughout history, it has been known by different names: white plague, consumption, phthisis, Shaya in Indian languages. And it was known to the Greeks as phthisis, as Shosha, Shaya, Rajashma in Indian medicine. Tuberculosis has been detected even in Egyptian mummies by modern techniques like PCR. So it's a very ancient disease. The victims of consumption or tuberculosis has been many throughout history. If you just take the literary figures, a lot of great writers, Guy de Maupassant, Chekhov, D.H. Lawrence, Orwell, Stevenson, and even our own Kerala, Changambura, John Keats, all were victims of tuberculosis. They all died very young in their 20s and 30s. So you can imagine the kind of devastation this disease has had in the previous centuries. It was really in the era of industrial revolution and industrialization, the kind of overcrowding and pollution which was there in the European cities of that time, that tuberculosis really flared into an epidemic of very large proportions. And it was very common to see hospitals like this or sanitaria full of tuberculosis patients, and the death rate was also very, very high. Charles Dickens called it a disease which medicine never cured, wealth warded off, or poverty could boast exemption from, which sometimes moves in giant strides, sometimes at a tardy sluggish pace, but slow or quick is ever sure and certain. Very depressing kind of statement. But in the last century and a half, from being a fatal disease, it has now become a curable disease. I would say in the aid of science. And you can see the picture of Medical College Thrissur, which used to be a TB sanatorium in the old days, now converted into a hospital because we no longer need sanatoria for this disease. And this story of the scientific intervention in tuberculosis, maybe you can say that it begins with Robert Koch's discovery of the tubercle bacillus in 1882. And steadily, the death rate due to tuberculosis declined over time to very low levels in Europe and much of the developed world. But even today, it is the number one killer among all infectious diseases. About 1/3 of the whole human population is infected. 8 million new cases occur every year. 3 million deaths occur annually. And now, looming as a great threat is the emergence of multi-drug resistant tuberculosis. This is the tuberculosis map of the world, and you can see that the dark blue is those areas with the largest affected areas, and it's mostly in Africa. And the next in list comes India and many other countries. The organism producing tuberculosis are mainly two: Mycobacterium hominis and Mycobacterium bovis. And this is an electron micrograph of the tubercle bacillus. And if you look at the structure of the bacillus, one thing that is very evident is the mycolic acid or lipid, a lot of lipid in its cell wall in the form of mycolic acids. And this is what gives it what the typical property of acid fastness, because the dye cannot be washed out away with acid, as we will see. The tubercle bacillus, once it enters the body, it can have different fates. In some people, those with normal immune function, the bacilli are completely cleared. So they do not develop the disease at all. On the other hand, there are a subset of people in whom, instead of clearing away the bacillus, there is a hypersensitivity reaction, and as a result, granulomas form in such people. And third are a group of people like those affected with HIV who have an immune defect, and so they develop generalized tuberculosis. And the initial infection produces a lesion known as primary tuberculosis, and primary tuberculosis can lead on to either a progressive disease directly or lead on to secondary tuberculosis, or the primary lesion can heal. This is what, of course, occurs in most people. Primary tuberculosis heals. That is the fate of most people. And primary tuberculosis can be asymptomatic, or there may be mild symptoms like fever, and if you take an X-ray, there may be hilar lymphadenopathy. So, the tubercle bacillus enters the lungs, and it enters the macrophages which are present in the upper respiratory tract and in the lungs. And these enter the lysosomes of the macrophages, and if no killing occurs because of the presence of certain factors like the cod factor and lipoarabinomannan in the bacillus or TB heat shock proteins, these are the things that prevent killing of bacteria inside the phagosomes of the macrophages. So in such cases, what happens is that these are processed and the antigens presented on the antigen presenting cells, and there are T lymphocytes, particularly the CD4 type of T lymphocytes, which are capable of recognizing this particular antigen, and this CD4 cells will elaborate interferon gamma, and this interferon gamma will activate or arm other macrophages into producing nitric oxide, and this nitric oxide will produce the killing of bacteria. So this is one thing that can happen in primary tuberculosis. So when this happens, this is what we call as immunity. Such people are immune to tuberculosis. They do not develop the disease. Now on the other hand, in others, interferon gamma can actually produce accumulation of macrophages, which sometimes alter in shape to form what are known as epithelioid cells. And these epithelioid cells will aggregate together to form what are known as epithelioid granulomas. And these granulomas enclose the bacilli. So the bacilli are in the midst of these granulomas. Then CD8 cells, the T lymphocytes, which are CD8 markers. So CD8 cells can recognize, capable of recognizing this, can produce cytokines which produce necrosis of tissue. And this necrosis in the center of the granulomas, a particular type of necrosis that we will see now later, known as caseation necrosis in the center of granulomas. So this is the typical delayed hypersensitivity. So a patient with tuberculosis can have two primary different fates. One, immunity or no disease, or delayed type of hypersensitivity, which leads to a granulomatous type of disease. Now this is a primary tuberculosis. In primary tuberculosis, the initial lesions are known as the gone focus, which is usually located in the small lesions located in a subpleural location. And also there are lymph nodes in the hilar hilar lymphadenopathy, and together these are known as primary complex or the gone complex. Now this is a picture of a gone focus, subpleural area. You can see that yellowish area there. And the hilar lymph node again, which has undergone necrosis, so caseous necrosis. Together, these constitute the primary complex. And histologically, primary complex is typical granulomatous reaction. See, what you see is all a granulomatous reaction. Epithelioid histiocytes mostly, and a Langhans giant cell. It's a giant cell with multiple nuclei arranged in a somewhat horseshoe pattern, periphery, arranged in the periphery. This is called a Langhans giant cell, very frequently seen in tuberculosis. And peripheral rim, around the granuloma, you find dark lymphocytes. So this together forms a epithelioid granuloma, which is typical of tuberculosis, and this is sometimes called a tubercle. So tubercle is nothing but a small epithelioid granuloma. And frequently, the center of these granulomas will undergo necrosis, a very amorphous eosinophilic kind of material that accumulates. This necrosis has been likened by its gross appearance to cheese-like appearance. That's why it's called caseation. Caseation and cheese-like. So this is caseation necrosis. And now what can happen to a primary complex? Now primary complex can go on to healing. So they just heal, and later on it can even undergo calcification. The lesions of primary tuberculosis, the gone focus and the hilar lymph nodes, both may undergo healing. And after healing, it can even undergo calcification. And these calcific spots can actually be detected on an X-ray. See, this is a hilar nodes, calcified hilar nodes in an X-ray chest. And histologically, it is just these tubercles or granulomas become heavily fibrotic, a process of healing, and they become calcified. All those blue areas are calcium. So this is a healed tubercle. Now the primary complex can also undergo another fate, though this is very rare. It can spread elsewhere in the lung, and this is called progressive primary tuberculosis. Spreading to the lung, and from a small lesion to a large caseous lesion, and this is destroyed caseous lesion of a progressive primary tuberculosis. Now primary complex can also spread via the bloodstream. This bacteria can spread via the bloodstream and form small tiny granulomas. And this is called miliary tuberculosis following a primary infection. Fortunately, this is also extremely rare. Now this is an X-ray showing what is called miliary mottling, small miliary lesions in the lung. They are called miliary because they resemble millet seeds. And this is a picture of multiple granulomas in the lung in miliary tuberculosis. And another fate that can occur primary complex is that it can spread to the brain. And this is one of the dreaded complications of a primary tuberculosis, though fortunately it is very rare. This is producing a tuberculous meningitis and CNS tuberculosis. And it can also spread to other organs. So this is tuberculous meningitis. Base of the brain you find an exudate, which is that of a tubercle, and you will find granulomas in such an exudate. And this is a meninges with granulomas and caseous necrosis from tuberculous meningitis. Now what happens to a primary complex? To summarize, it can undergo healing and calcification. It can progress on to a progressive primary complex, or it can produce a miliary spread, or rarely tuberculous meningitis, or spread to other organs. The last three are fortunately very uncommon. Apart from the lungs, primary complex can also occur with the initial gone focus or the focus in the primary focus in the intestine in case of Mycobacterium bovis, or sometimes in the oropharynx. Sometimes the primary lesion may be in the skin, or even rarely in the liver in case of a transplacental infection in which a baby's liver may have a primary complex. Now coming to the most common form of tuberculosis, the most common form of disease, that is secondary tuberculosis. Secondary tuberculosis can occur either as reactivation or due to reinfection. Reactivation means the bacilli in the primary complex are already lying dormant and they become reactivated. Reinfection means the patient gets a infection all over again after the initial primary complex. Secondary tuberculosis can manifest as fever and cough. These are the most common manifestations. Sometimes when it progresses on to cavitary lesions, it can produce hemoptysis, and weight loss and a raised ESR are very frequent manifestations of secondary tuberculosis. Now those cases of secondary tuberculosis, if you inject a tuberculin antigen onto the skin, it will produce a typical indurated swelling reaction to tubercle antigen, and this is how it looks like. And when it measures, it is indurated, large lesions are present. We say that the tuberculin test is positive. Now, in those cases of healed and calcified primary complex, sometimes bacteria can lie dormant within these lymph nodes or the primary focus, and they can become active once again, and these bacteria may then go on, because tuberculosis is the typical aerobic kind of bacteria which prefers areas of high oxygen. So it goes to the upper lobes frequently of the lungs and establishes granulomatous lesions of tuberculosis. This is the classical secondary tuberculosis. And another thing that can happen is a patient who has already had an infection once can be reinfected by a secondary a wave of bacteria coming from outside. This is reinfection. Again, these bacteria can go and settle, preferably in the upper lobe of the lungs, but also it could be anywhere else, and set up a secondary tuberculosis. So, two ways in which secondary tuberculosis can occur. And these lesions of secondary tuberculosis can sometimes cavitate, become cavities. And these cavities can get attached to a bronchus, and through this route, bacteria can go out again when the patient coughs or sneezes, and this kind of bacteria may infect other people. Such cases which actively shed bacteria are called open tuberculosis. So these are open cases of tuberculosis, and this is the main source of tuberculosis infection in a community. And that is the importance of testing the sputum to look for bacteria, is to look for open cases of tuberculosis. And sputum examination is done mostly by a Ziehl-Neelsen stain. Ziehl-Neelsen is an acid-fast stain in which the dicarbon fusion cannot be washed away with acid, unlike the usual bacteria. So it retains the dye, and this is called acid-fast. See, look at the long rods of tuberculosis. Another way is to look with fluorescence microscopy using an auramine stain, auramine or rhodamine stain, and here you can see this bacteria with a fluorescence microscope. Now, a cavitary tuberculosis can spread from that cavity or the primary, the secondary focus. It can spread elsewhere in the lung, and in such cases you will call this progressive secondary tuberculosis. These lesions, this is a cavity in a chest X-ray. This is progressive secondary tuberculosis in both lungs. This is a massive involvement of one lung. CT scan again to show a very large cavitary lesion. And this is the initial stage where it spreads to the rest of the lung. It can present as what is known as a tuberculous bronchopneumonia. And these lesions can enlarge. You can see the several caseous foci in this lung, and can completely involve a segment of the lung and become very destructive and fibrotic later on. This is a cavitary lesion, upper lobe cavity in a lung. These are the granulomas in the lung with caseation. Miliary tuberculosis can also, far more frequent than in primary tuberculosis, miliary tuberculosis can occur in secondary tuberculosis. From the secondary focus, see in that upper lobe, it has spread via the bloodstream into rest of the lung. This is the miliary mottling of secondary tuberculosis, miliary tuberculosis. A CT scan showing the same. Small nodules are seen. This is lung tissue showing miliary mottling. All the small millet-sized small areas are all miliary tubercles. Higher power to show this miliary tubercles. And this miliary TB, the miliary tubercles can be picked up even by fundoscopy. By looking at the patient's fundus, frequently you can find this miliary tuberculosis. That's one good way of diagnosing miliary tuberculosis if you suspect that in a patient. This is liver again showing miliary tubercles. Secondary tuberculosis again can spread to other organs from the lungs to the brain or into other organs, just like progressive primary complex. This is intestinal tuberculosis showing ulcers in the intestine, and these ulcers are frequently circumferential ulcers, and these ulcers can be long, circumferential, and they can even lead to chronic disease and strictures of the intestine. So this is one of the important causes of strictures because of fibrosis of circular lesions, circumferential lesions. They become stenosed or they form strictures, and this is can be a cause of intestinal obstruction. This is microscopy of an intestinal tuberculosis. Lymph node tuberculosis. Lymph nodes, particularly in young patients, this is very common, and previously this used to be known as scrofula. Scrofula used to be a very common form of tuberculosis in the ancient times. Both intestinal and even scrofula could be due to Mycobacterium tuberculosis or bovis. Intestinal tuberculosis Previously, Mycobacterium bovis used to be the common cause, but now it is Mycobacterium tuberculosis because bovis has become extremely rare, and Mycobacterium tuberculosis, because of swallowing of sputum from secondary lesions, is a more common cause. Kidney can be affected to form a tuberculous pyelonephritis. You can see the pelvis completely filled with caseous material. This is a tuberculous epididymitis. So it could be a cause of male infertility, loss of strictures and other fibrotic lesions in the epididymis, tuberculous epididymis. Similarly, in the female, it can produce tuberculous salpingitis, another cause of infertility in the female. Apart from meningitis, the tuberculosis can produce tumor-like lesions. And these are called tuberculomas because they can be mistaken for space-occupying lesions, some other tumors. So in India, when you see a space-occupying lesion in countries like India, tuberculosis is a very important differential diagnosis. This is tuberculous laryngitis, another form of presentation. And this is tuberculous osteomyelitis. You can see the vertebrae here, which are affected and destroyed by the TB. And it can produce lesions, even paraplegia. So this spine affected by tuberculosis, it used to be a very common disease once upon a time. Fortunately, no longer that common. This used to be called Pott's spine. And it could even produce destructive lesions which can impinge on the spinal cord and produce paraplegia. And it can track down into through the vertebrae into other spaces and produce typically like a psoas abscess. This is also another presentation. So you can see that tuberculosis can affect virtually every organ, producing various kinds of manifestations, so much so that it can mimic so many other diseases. And whichever organ, whatever disease, frequently, that is why tuberculosis becomes one of the differential diagnoses that we have always got to consider. In case of HIV infection, tuberculosis and HIV infection frequently go together. And HIV infection all over the world, tuberculosis is one of the consequences. Because as the CD4 count comes down to a particular level, they tend to contract tuberculosis. That's why there's been a resurgence of tuberculosis in many countries, especially in Africa, because of this HIV infection. And unlike the usual primary or secondary tuberculosis, these lesions, because there's no immunity, there's very little granuloma formation, because granuloma formation is a delayed type of hypersensitivity which needs intact T cell, CD4 kind of T cell to form these granulomas. So there's no granuloma formation, and the consequence is that the bacilli simply accumulate within the macrophages, like in lepromatous leprosy. You remember the lepromatous leprosy where the foam cells accumulate bacteria. Similar to that, this can accumulate in the macrophages. Like in this picture, see, these cells are inundated absolutely with thousands, millions of bacteria. So, tuberculosis is a protean disease, can produce multiple symptoms: cough, fever, hemoptysis, abnormal X-ray, pleural effusion, malabsorption, intestinal obstruction, diarrhea, mass abdomen, lymphadenopathy, seizures, coma, paraplegia, arthritis, infertility, etc. All this, tuberculosis forms a differential diagnosis. Now, a little bit about the pathogenesis. All persons infected with Mycobacterium tuberculosis will not have the same fate. So what is the reason? These people can have either no disease, mild disease, or severe disease. So what is the reason? Here we have to consider both host factors and agent factors. Host factors, namely nutrition, genetic factors, and co-infection with HIV. These are the important things to consider. And as Regarding agent factors or the tubercle bacillus itself, the virulence of a particular strain of bacteria or the bacterial genes can vary from strain to strain. So both these are important. Coming to the host factors, there's a genetic variability in human susceptibility to tuberculosis. And the severity of disease is generally greater in America, for example. The African American population infected with tuberculosis have a more severity of disease than the Anglo-Saxon or the white population. There are some groups like the Yanomami Indians of Brazil. It is estimated that 6.4% of the population has active tuberculosis. So some populations are very susceptible to the disease unlike others. Now, what is the reason for this susceptibility? There are multiple genes. Many genes are now being investigated by all kinds of molecular genetic studies, particularly the so-called genome-wide analysis or GWAS kind of studies, looking at multiple genes and for susceptibility as well as severity. Now, one example is the NRMAMP1 gene, otherwise natural resistance associated macrophage protein 1. This is a gene found in chromosome 2 in humans, and it is thought to be a nitrate transporter. And several polymorphisms of this gene are actually found, and some of these polymorphisms confer more susceptibility to the disease, and some have more resistance. So it's important in multiple populations to study not only NRAMP but many other such genes in different populations to know which confer more susceptibility, which confer more resistance. That is one way of investigating the molecular pathogenesis of the disease. Now, regarding agent factors, some mycobacterial strains grow extraordinarily rapidly in mice. So is it associated with bacterial gene polymorphism? This is again another thing that is being very actively looked into. And one of the things that aids us is that, like the human genome, the Mycobacterium tuberculosis genome is also now completely decoded. We know the entire Mycobacterium tuberculosis genome. And this is the Sanger Institute Wellcome Trust site, where if you go to that site, you can look at have a glimpse of the entire Mycobacterium genome. And the different protein and DNA sequences can be found out from this genome. Now I will conclude by reminding you once again that tuberculosis is one disease that has become curable from what used to be an incurably fatal disease. There was a time when a diagnosis of tuberculosis was like saying that now that patient has some severe kind of cancer, because most likely this patient was going to die. But now it has become a curable disease. So for the future generations, we can look forward with great hope regarding this disease. But in order to do that, we also have to solve a lot of problems. Particularly, see, this is a view from an aeroplane as it lands in Mumbai airport. You can see the large skyscrapers, and along with that, the shalls or the slums that you see where there is so much of poverty and overcrowding, which are the typical breeding grounds for tuberculosis. So the social conditions also have to be looked into, and we can successfully use science to overcome this disease only, even by looking at or solving the social problems as well. Thank you.